RNA purification affinity method. This is another method by which we can purify the RNA from different cells and from the tissues. During this procedure, cell is lysed. The cells that contain the DNA or the RNA, they are lysed and spin to remove large particulates or the cell debris. During the first step, they are spun or they are centrifuged after the cell lysis. The first step should be cell lysis. Once there is cell lysis, then they are centrifuged to remove the large cell debris. Once large cell debris they are removed, then the lysate. Lysate is that solution in which there can be DNA and there can be RNA, which is present currently in the solution. So containing nucleic acid and cellular contaminants, they are joined to a column. That column is of glass membrane. So during this, because the name of the protocol is affinity method, during this method a column is used with a glass membrane. That column has a glass membrane. Wash the sample with alcohol to remove the contaminants. Nucleic acid stick to glass membrane. Both it can be RNA. They stick to the glass membrane which was present on the column, while the contaminants they wash off. So during this process, the RNA that is present in the sample that stick with the glass membrane which was present on the column. Due to this stickiness of the RNA to the glass membrane, this method is called as affinity method. In the next step, treat the DNase enzyme, because there can be RNA, there can be DNA, because both DNA and RNA they stick to the glass membrane. So DNase enzymes they are used. So these enzymes they remove the contaminating DNA, or these enzymes they degrade the DNA. So once the DNA is removed or the DNA is degraded, apply water to the column. Because RNA is still attached with the glass membranes, but the DNA has been degraded or it has been removed by the enzymes which are called as DNases. So after applying water to the column, purified RNA washes off from the glass membrane and it is collected in a tube. So in this method, we can use the DNase. Once the RNA or DNA they attach with the membrane, later on we use DNases. With the help of DNases, DNA is degraded or it is removed from the membrane, and by using the water we can remove the RNA from the glass membrane, and that can be collected in a tube. So this is the method that is called as affinity method. Affinity method is also used for purification of the RNA. So there are certain advantages of this method that is called as affinity method. In this method, no use of the organic solvents. There is no use of chloroform or phenol. We can use inorganic solvents. Variety of samples we can use and can purify RNA from a variety of the samples. They can be tissue or the cultured cells. They can be white blood cells. They can be plant cells, or even we can purify the RNA from bacteria and yeast. There are some other advantages of this method that can be DNase, which we use during this process. They degrade or they eliminate the DNA which was present in the nucleus of the white blood cells. In addition, this method produces RNA that have excellent purity as well as integrity.